Well, I want to sort of transition our discussion a little bit to some of the operational implications of quality. In the past couple of months, we've talked a little bit about some of the regulatory pathway considerations, some of the pathways that the leaders have carved for us. Right, we have products in the market that kind of serve as an example for us. But I would also say there have been a few real-world examples in the past that emphasize some of the difficult trade-offs, right, that we have to face in quality right now as it relates to, for example, perhaps achieving the scale and quality that we need, and particularly as we're transitioning, right, from early development to later stage commercial ready development. And some of that, I think, is just linked to some of the immaturity that we're facing in terms of platform development and technical immaturity. So I want to take our discussion a little bit to some of the upstream and downstream considerations, and best practices that you guys have utilized or are looking at, and then look a little bit too into the outsourcing world and thinking as well about comparability as we're scaling up, right, or moving forward with our products through the clinic. So let's start, Justin. I'd love to understand how you're approaching, upstream process development. Where do you see more room for growth or improvement in our upstream development, and what kind of knowledge do we still need to learn, or process-related hurdles do you think we're really facing as an industry to help lighten the lift, if it were, right, as we're moving towards downstream purification?
Yeah, it's a great question. I mean, we've been spending the last 8 years I've been here, trying to move to continuing to increase productivity, continue to increase control of our upstream manufacturing process. A lot of the early ways are moving away from an inherent technology into a suspension-based technology. Some have, obviously been able to scale out the adherent-based technology, but I think, we've transitioned from the adherent to suspension, and then we've continued to try to innovate within that space from, trying to increase looking at the cell line, the cell clone you're using, and productivity of your cell clone, the efficiency of your transfection, and how many cells are you able to reach during your transfection reaction, and then how much you can push those cells to produce vector, or similarly, can you improve packaging within them to reduce the burden on your downstream process? I think those are a number of the things that you can, it's obviously art to that you go through, but I think those are largely a part of it. And I think generally, if you're looking at, gene therapies reaching beyond the rare diseases, or especially to neuromuscular conditions, you need access to larger scales. You need to be able to be more productive, and, higher purity coming out of your upstream process is highly desirable from a yield perspective, and also from a end product quality standpoint.
Absolutely. I think there was there's been sort of the debate in the field of, the SF9 versus HEK293, right, cell line development. And there were some big papers, I think, just recently released looking into that, right, and revealing some answers that were maybe hard for some folks to hear. So it's one of those. Justin, you blinked. I'm not sure if that was a.
No, and I think each company is kind of, you pick your platform and you look to get very good at your platform, and it takes a lot of innovation to have the true understanding of what your platform can do and what the quality attributes that it produces from it. So I think that the industry is not fully harmonized on a platform at this point, and some of that depends on the serotype you're producing. And some of it depends on the disease indication you're going after, or also the technical capability of your staff, to be frank, right, where they have their experience. So Regenx is, we're a suspension-based triple transfection, and we've spent the bulk of my time here innovating that space, and have really increased our understanding and our capability within it. Others have done similar with other technology and expression systems.
Yeah. Do you feel, I just want to add on a quick question there, do you feel that we will arrive at one standardized cell line platform, or do you think as an industry we will be able to juggle, and our outsourcing companies, right, our partners, will be able to handle that?
I mean, I think the dream is a producer cell line at some point that you can harmonize around like a monoclonal antibody, but that's been a long path coming, and that's probably, I don't think that's in the near term time horizon, but maybe in the 10, 20-year horizon, maybe that becomes more, the science is there and people are able to harmonize around that. I mean, you saw monoclonal antibodies harmonize around that, over time. We're still in the earlier stages of, I guess I'll put it this way. When somebody lands a platform that fully, truly outperforms all the other ones, it will force harmonization because of that.
That's a really good point.
We're still in the little bit of the bake-off stage, maybe.
I used the term bake-off in an article earlier this month, and it makes me laugh. I love that because it is. That's exactly where we're at. Andrew, go ahead.
I was just going to say it's very accurate. And I saw we had the question from Rihanna in the chat about producer cell lines. So it was we just discussed as are they just another alternative to transient transfection? So it's interesting. I agree with Justin that in the medium to long-term time frame, that producer cell lines will probably be the way forward. If you look at the complexity of designing a producer cell line for an AAV, which is basically toxic for the cells, and then compare that with, say, a traditional producer cell line where you stick one gene for one protein, and it's like a log scale increase in complexity, and you have all these cascades, and you have to think about the helper-virus, how would you use of that? And I know Ultragenyx has the Pinnacle system, and when they have kind of a producer cell line, but they do use, I think, Ad5 as a helper virus. So they would say kind of that they're already there, but then you can argue that having Ad5 isn't desirable from an operator safety and it's a little bit more complicated for the operators, and then you have to show the viral clearance, which is very doable, but it's just these are kind of negatives in their ideal world you wouldn't have. So
interesting.
Producer cell lines, but otherwise I kind of agree with what Justin said, and we're also transient transfection suspension cell line based right now. I think for some of these like orphan indications where it's very low dose, just imagine an I dose or an EA dose with Regeneron's acquisition of Decibel. I think you could use transient transfection and it would be fine. It would be super quick to develop in the future, and you would be able to easily make enough for the very low dose, low number of patients as you need.
I think your expression system depends somewhat on the disease and what you're going for here. Something that's rare diseases are much more amenable to fast translation technologies, whereas something that is a larger, you need a larger amount of vector either for an individual patient or a total population. More upfront development may be beneficial so that you are able to produce at scale over the longer term.
Well, and I think too, that's one of the bigger questions we as a field are facing right now, right? It's currently a lot, probably a majority of us are working in the rare disease spaces, right? And then there are other companies that are trying to sort of bridge the gap, right, between the rare diseases into the larger patient populations. And I think a lot of it, we're still, just as we're trying to figure out our AAV, we're still trying to figure out how you can actually move these products into a broader patient population. And that's going to, I think, have larger implications as well on cell line development and your upstream process and what ultimately wins out in terms of, producer cell lines and harmonization there, right, if there is any. I would like to kind of further this conversation a little bit on the producer cell lines. I'm just really curious to get your sense, what innovation do you think would actually make that a reality? Is there there's probably not a single innovation that would help us get to that point. But, if you had to sort of give us some brain cells around where you see innovation happening or could be happening to try and further bring us closer to that goal as an industry, what would you think should be a priority? And Justin, we can start with you. And then Andrew I don't know, he looks like he's got a devious grin on his face, so he may have a good answer.
I think it comes down to the understanding of cell biology and how you can take this highly complex system and truly turn it into a stable producer cell line. I think there's been a lot of work around there. There's even technology out there that are the initial stages of producer cell lines coming into that. So it's probably I'm out of my depth. I'm not the at the micro level on these. I tend to stay at the macro level of production. But it's really solving that, I think, biology question of how you can program these cells and keep them from dying effectively to produce what you want. That's the innovation. I think there's been bites at the apple and we're getting closer, but that you know, I haven't seen us there yet.
Yeah. Yeah. Andrew, what are your thoughts?
I was just going to say, I don't think it's one innovation is going to do it. I think you need it's like playing Space Invaders, where you have like 50 different things coming at you. You just need to blast each one by one until you get closer and closer and closer. But you've got the stamina to be in the game for long enough.
Oh my gosh. I love it. Space invasions, guys. We've got space invasion, space invader analogies coming to stage here. There's beautiful videos that we could come up with for that.